After discussing gRNA, now let's concentrate on small silencing RNAs. These three are small silencing RNAs. Small silencing RNAs. siRNA, miRNA and piRNA. Now let's discuss about what are small silencing RNA. Small silencing RNA, as the name suggests, it will be a small RNA of almost 20 to almost 30 nucleotides long. Okay, and make an association with the members of Argonaut protein family. Argonaut protein family, which they guide to their regulatory target. After the discovery of small silencing RNA in the year 1993, discovered of small silencing RNA was in the year 1993. After discovered, several small classes have been identified about small silencing RNA, which differ in their biogenesis, their modes of target regulation, and in the biological pathway they regulate. There are three types. The full form of siRNA is small interfering RNA, then miRNA, full form microRNA, and piRNA, full form PV, interacting RNA. Okay, so these are three types of small silencing RNA: siRNA, miRNA and piRNA. Small interfering RNA, microRNA and PV interacting RNA. Now let's discuss about miRNA. miRNA. They are endogenous and obviously regulatory RNA. Then 20 to 25 nucleotides long. Next, thought to regulate the expression of other genes. miRNA derived from pre-miRNAs. Primary miRNA derived from this pre-miRNA. Pre-miRNA to miRNA. Next, transcribed by RNA polymerase 2 enzyme. Next, the pre-miRNA. Now let's discuss about the processing. The pre-miRNA inside the nucleus. 60 to 70 nucleotides long miRNA by the activity of DROSA. And what is this DROSA? This one is a nuclear enzyme. The pre-miRNA. Note the spelling. This one is miRNA. In the nucleus, this miRNA is modified into 60 to 70 nucleotides long pre-miRNA, and this is formed by the activity of DROSA. DROSA is nothing but a nuclear enzyme. After that, what happens? First pre-miRNA to pre-miRNA. It occurs into the nucleus, and then pre-miRNA come out of nucleus. Okay, out of nucleus actively, and come out very actively, and come into cytoplasm by exporting protein. Now come to another interesting term, the dicer enzyme. The dicer enzyme, a dsRNA specific enzyme. RNAs3 form RNAs3 families. And then cuts pre-miRNA into mature miRNA. This is the whole scenario about first pre-miRNA, which resides in the nucleus, change into 60 to 70 nucleotide long pre-miRNA. See the spelling difference, PRI and PRE. And this one is happened by the activity of DROSA. DROSA is the nuclear enzyme. Nuclear enzyme as it is done into inside the nucleus. So this nuclear enzyme DROSA, by the activity of this nuclear enzyme DROSA, the pre-miRNA changed into 60 to 70 nucleotides long pre-miRNA, and this pre-miRNA then comes out of the nucleus actively into the cytoplasm, and this actively transport is helped by the exporting protein, and this in pre-miRNA comes out of the nucleus actively into the cytoplasm with the help of exporting protein. Then come to another concept, the dicer enzyme. Dicer enzyme is a dsRNA. Make this not dsDNA. This dsRNA means RNA double-stranded RNA specific enzyme, which is under RNAs 3 family endonucleates, then cuts, and this dicer enzyme, it cuts pre-miRNA into mature miRNA in the cytoplasm. Okay, so this is in a nutshell miRNA. Okay, after discussing miRNA, now let's concentrate about siRNA, another small silencing RNA. That one is siRNA. Form of siRNA is small interfering RNA. Small RNA molecules of almost 21 nucleotides. siRNA duplexes are produced by dicer. We have already known about dicer. Dicer is a dsRNA enzyme, 2 strands. This duplex, out of these 2 strands, one directs silencing of target mRNA is called guide RNA. The other strand out of these two strands, the other one is ultimately destroyed. It is known as passenger strand. This target mRNA, target regulation is done by RICS. And what is the full form of this RNA induced silencing complex. siRNA may be exosiRNA and endosiRNA, depending on the source of RNA. So let's summarize siRNA. siRNA full form is small interfering RNA. It is one of the small silencing RNA family. siRNA, this is a small molecule of about 21 nucleotide long. siRNA duplexes are produced by dicer enzyme, as we all know from miRNA studies. The dicer, this is an dsRNA enzyme of RNase 3 family, and this is endonuclease enzyme, and out of the duplex, siRNA duplex, and out of this two strand, one direct silencing of target mRNA is called guide RNA, okay, and the other strand that is destroyed is also called passenger strand, okay. The siRNA, the target protein, here the target mRNA, the target regulation is done by RISC, and the full form of RISC is the RNA induced silencing complex. The siRNA is of two types, siRNAs are of two types, exosiRNA and endosiRNA, and these two types depends on the source of RNA. So this is all about siRNA. Now let's about the third point, piRNA. We have no much information, data about piRNA, only the full form, the PUE interacting RNA. piRNA, PUE interacting RNA.